Every organism is constrained by space, because at any given moment, we're always occupying a physical space as well as completely surrounded by space. And so our success in terms of reproduction and survival rely on our ability to navigate space effectively and efficiently. And that goes for every organism. Now, for the organism that I study in particular, it's the prairie vole. And across sort of the top banner, I've outlined the traditional way that people study prairie voles. They're used to understand monogamy. So typically you look at the genetic components that then impact the hormones in the brain, and then how that expression impacts behavior. And the behavior I'm depicting there is two voles falling in love. But really, I want to take monogamy and really deconstruct it and bring it down to its spatial components. Because at the heart of it, monogamy is inherently spatial. The ability to be able to find a mate, to find resources, and then defend that territory and guard those things that you've gathered requires a high level of spatial capacity. And so I test just that in the lab using what's called the Morris water maze. So it's a big tank of water with visual cues and then a hidden platform. And these two pictures below are showing actual data that I've collected, track visualizations from animals. And so what I'm trying to show here is that you can assess how quickly and efficiently an individual can find that hidden platform. And I have two different individuals modeled for you. One on the right that was really great at finding the platform, and one on the left that was not so great. But what's really interesting about this is with thinking that traditional pathway of how we understand hormones and understand how the brain is impacting love or monogamy, some of those same hormones like oxytocin that are important for, say, aggression or pair bonding are also showing up here to understand spatial memory. So in fact, I was able to show that oxytocin is expressed in the hippocampus, the picture here in the middle, which is almost exclusively an area of the brain studied to show how spatial learning and navigation come about. So this is great evidence to show that you can actually link the way we traditionally think about different behaviors or mating systems, and we should really be looking at them with a spatial lens. And what I was able to show is that individuals with high levels of oxytocin in their hippocampus were actually really bad at the water maze, where individuals with low levels of oxytocin in the hippocampus were much better. And so how do all these things relate together? And does spatial ability actually dictate the realized mating tactic of an individual? So for that, I'm taking my animals from the lab out into the field. And so what I can do is actually test their ability and track them in real time and look at how they use space, if they're exhibiting monogamous mating patterns, and also look at their reproductive success. So using this really integrative approach of combining the ecological relevance of spatial learning and navigation and memory, but also the neural mechanisms and its underpinnings, can really show great insights into not just monogamy, but other mating behaviors, and really any animal behavior that's observable in the natural world. Thank you.